Hey everyone, thanks for joining into my talk. My name is Meirav, and today I will discuss how we can use data science for biological research. So, quick hello. Hi, I'm Meirav. I'm a data scientist at Authorize. And back in the past, I did a master's degree in bioinformatics where I focused on the microbiome. So, I analyzed many data sets, I answered many biological questions, and I also developed algorithms for research of the field. In my spare time, I write blog posts about data-related topics, which you can find on Medium. And fun fact, I used to have a podcast about science where I interviewed other researchers about their work. So what are we going to do today? First, I want to put us all on the same page in terms of terminology, so I will explain what the microbiome is and why it is so interesting and important. Then I will show and explain how we represent the microbiome numerically, so we can apply data science on it. Then I will show one cool thing we did with data science on such data sets, and eventually I'll wrap everything up and show how we go from data science results to biological insights. So what is this microbiome thingy you were so obsessed with, my love? Well, according to Science Magazine's definition, the microbiome is the entire collection of microorganisms in a specific niche. And by niche, we mean a specific location. And that could be anywhere you can think of. It can be an organ in the body, and it can also be exterior, for example, a spot in the desert or the sea. So just to give an example, I once interviewed this researcher who studies the ocean, and he told me that one way for him to know what's going on in the depths of the sea is to look at the bacteria that live there and compare it to bacteria that used to live there 1 year ago, 2 years ago, maybe longer ago, and that gives him a decent idea of what's going on in there. But the sea is very far from us, so let's stick to our bodies. And in our bodies, it is known that the bacteria is present in every organ and they're involved in nearly every possible biological function you can think of. So just to name a few, we know that there is microbiome in the brain and they're involved in many interesting things there. For example, they mediate hormone secretion, which influence how we behave and feel. They are involved in obesity and metabolism. They're involved in immune response and allergy. They're influencing pregnancy and also fertility. They influence how the body responds to cancer, and they have also been tested as a way to treat cancer. And last but not least, they also affect our mental state. So depression, anxiety, bipolar disorder, all those things are related to bacteria and the other way around. So what I need you to remember from this is that the microbiome is the microbial community in a specific location. So we're all convinced by now that the microbiome is important and interesting, but we also know that to do data science, we need a table with numbers, right? So how do we get from a bunch of bacteria in the body to a table with numbers we can feed our algorithms with? Well, in one word, sequencing. And the pipeline is as follows. First thing we do is we take a sample from the niche we're currently exploring. So if we're talking about the sea, we will take a bit of water. And if we're talking about the mouth, we'll take a bit of spit. Then we'll do DNA extractions, which means that from each sample we collected, we will pull out all the genetic material we can find in it. Then we will do sequencing. So a small reminder from your biology class, DNA is a double-stranded structure, which is made from nucleotides. And we have 4 types of nucleotides, which we represent with one of the following letters, A, C, T, or G. And you can imagine the DNA as a necklace, where each bit is a nucleotide. And when we sequence it, we basically put it through a machine that can tell us per each bit which of the nucleotides it is, and it also writes it down for us. How convenient. So the output of this phase is a text file with a sequence for each of the bacteria we found in our sample. But one thing the machine doesn't know to tell us is to which bacteria the sequence belongs. It can only tell us the nucleotides. Well, for that, we have a database with many sequences of bacteria that the scientists already knew and they sequenced. And in the database, for each known bacteria, we have its sequence and its name attached to it. Again, convenience. So what we do now is we take each of the sequences we found in the sample and we compare it to each of the sequences we have in the database. And once we find one that is over some percentage similar to the sequence we have, we say that our sequence is of that type. So eventually we get this beautiful table where each column represents a bacteria and each row represents an individual from whom we took the sample. And each cell represents the concentration of the bacteria inside this individual. Very easy. So now that we have data, let's do some data science. So how many of you know what mucositis is? Very good. So I hope today will be the first and last time you hear about this. Mucositis is an inflammation that results in wounds inside the mouth. And everyone that goes through bone marrow transplantation gets it. And an interesting thing about it is that there is a big diversity in how patients experience this inflammation. So after the transplantation, we go to patients and we see in each of their mouths wounds. So we know that everyone has it. But when we ask them about it, some of them say that they can hardly feel it, and some of them suffer so badly that they can hardly speak or eat. Now, there is medication you can give in advance to stop this from occurring. But one thing, it costs money. And second thing, usually the doctors prefer not to give it because the body, when it's going through transplantation, it's already overloaded, right? It has cancer. The transplantation is very weakening. It's been treated with other things at this point. So the doctors prefer not to load it anymore. And they also say that if I don't know which of the patients will suffer, then I prefer to not risk it and just not give it to anyone. The worst case, some will have a bad time. But what if there were tiny little creatures that could tell us in advance, even before the transplantation, which of the patients will suffer from mucositis really bad and which won't? So we know who to give the medication to. So luckily, they're called the microbiome. And long story short, at some point of my master's degree, I was asked to see if we can use the microbiome in the mouths of patients before the transplantation to predict how bad they will suffer from the mucositis after. So we went to the patient, we took a sample from their mouths, we extracted the DNA, we sequenced it, and we found this beautiful table. Then after the transplantation, we went back to them and we asked how bad do they suffer from the mucositis. So for those who said they suffer badly, we labeled their sample as 1. And to those who said they hardly feel it, we labeled the sample as 0. And after some efforts, I managed to build a classifier, a random forest, if you're really interested, that could do the prediction pretty accurately. So congratulations, a POC is born. We can say who will suffer from mucositis real badly and who won't. So this is an example of something cool and meaningful we can do with data science applied on microbiome. But this is pure data science, and I promised you biological insights. So let's go back to biology and see how we go from the data science results to new insights. So if you remember when I showed you the table of the microbiome, I'll show it again now, you can see that our features are basically the bacteria. So once I have a classifier to distinguish between, for example, sick and healthy patients, then I can use feature importance to extract a list of the bacteria that was meaningful to the model's decision. And I'll take this list of bacteria and I'll go to the literature and I'll see if I can cross information between what is known on this bacteria and what is known on this condition to learn new things about both or maybe only the condition. So to give a practical example, let's imagine that I have a data set taken from Crohn patients. And some of them are suffering from flare, which means that their Crohn is losing it, they suffer from its symptoms. And some of them don't, they don't feel the Crohn at the moment. Now let's imagine that I built a decision tree to distinguish the sick from the healthy, and that in this decision tree, I found out that for some bacteria, when its levels are over 43, then the patient is sick, and when it's lower than 43, then the patient is healthy. Let's also imagine that I took this bacteria and I went to the literature and I learned that this one thrives on glucose. So one easy conclusion would be to say, well, dear Crohn patients, if you don't want to suffer flare, then you might want to go easy on the sugar, right? Because when you eat sugar, you nurture this bacteria directly, its levels rises, and you suffer from attacks. And of course, this is very basic. I ignore many complexities. I ignore dependencies between the bacteria. I ignore also causality, just to make the point clear. So that's what would happen if life was really simple. Another thing that could happen when I went to the literature is that I found that this bacteria doesn't necessarily thrive on glucose, but it does secrete some compound that contributes to the attack, or that it depends on some bacteria that does something else that influences the attack. So this is a bit abstract, but the thing is that sometimes when we go to the literature, we learn that the bacteria is involved in some biological process that affects the condition we learn about now. And this is teaching us something about the mechanism of the condition we're studying. And that's how new insights are born. And that pretty much wraps up everything I wanted to say today about the microbiome. I'm free to answer questions if you want, and please feel free to email me if you think about anything later. Thank you so much for your time. I hope you find this valuable, and let's be in touch on LinkedIn. Thank you.